Good morning, everybody. Today we're doing chapter 3A, and if anybody shows up, review for this first exam. Good morning. How are you doing today? Can you hear me all right? Chat, yes. Yeah, I'm tired too, but I appreciate you coming. It's nicer to have somebody in than nobody. Okay, it's now 9 o'clock, so I'm gonna go ahead and get started. Anybody else shows up and we will add them in. Today is chapter 3. I hope you have your notes printed, ready to go. Okay, chapter 3. First thing we're going to be talking about is the cell theory. What is it? Cells were discovered in 1666 when the first microscopes were invented. This was when they were first able to grind lenses good enough to be able to see both at a distance out into space and down into the microscopic world. When better microscopes were invented, that means the ability to grind the lenses better and to add several lenses together on top of each other. Smaller parts of the cell called organelles were discovered. And by the way, stop me if you have any questions as we go. Examples of organelles are the nucleus, mitochondria, chloroplasts, lysosomes, and Golgi bodies. That's a small list. There's more than that. All living things are divided into two groups, whether their cells have organelles or not. Okay. Now remember, you may not be able to fill in your notes as we go, but you can definitely fill it in from the PowerPoint and from rewatching the video. Okay, this is a picture of a prokaryotic cell. You can see some of the cell parts there. What they don't have in this picture is organelles. Prokaryotes have no organelles. This is an example of one, bacteria. Bacteria, eukaryotes like us, they do have organelles in their cells. Example, plants and animals. So there's animals and plants. By the way, not only are these all animals, they're also all mammals, but there are a lot of non-mammal animals, like insects and fish, things like that, that are also animals, including people. Okay, prokaryotic cells. You can see the prokaryotic cell in the upper left. It has what's called the nucleoid region because it doesn't have a nucleus. It's much smaller, it's less well organized, less complex. Notice the eukaryotic cell has a nucleus and organelles, much larger, much more complex. Okay, there's that picture of the bacterium again. You can see some of its external structures such as pili, flagellum for swimming, the chromosome, DNA, and cell wall. They're always smaller than eukaryotic, almost always smaller. They lack internal structure surrounded by membranes. So that's the main difference between prokaryotic and eukaryotic cells. And they don't have a nucleus. So even though they have DNA, they don't have it surrounded by a nucleus. So there you go. They can have flagella or other external features such as pili, a capsule, and a flagellum. Okay, eukaryotic cells like us. They're usually larger than prokaryotic cells. They possess internal structures surrounded by membranes, which we call organelles. They possess a nucleus, and they can also possess flagella or other external features. Okay, this is from your text. This shows you the different organelles and partly what they do, a little bit of what they do. So you should probably learn these things and have these ready for the exam. This is a plant cell showing you the structures they have. They have slightly different structures than we do. For example, they have a central vacuole. They have chloroplasts, which we don't have. All living cells, what do they have in common? Well, both prokaryotic and eukaryotic cells have at least these three things in common. The first is you got to have a cell membrane that surrounds the outside of the cell. It's selectively permeable, which means it can let some things in and not others, and vice versa on the outward movement. DNA, all living cells have DNA to control cell activity and code for proteins and for inheritance. And the cytoplasm is the liquid part of the cell. All living cells have these, but most living cells have what is called a cell wall. Now, animal cells don't, believe it or not, animal cells are not most of the cells on the planet, but most prokaryotes and eukaryotes that are not animals have a cell wall that supports the cell and prevents it from rupturing. Okay, there's an image from your text showing how your cell membrane is set up. It's composed of a phospholipid bilayer. So if you look at this figure, the phosphate part, the phospho part of it is the red ball there, that's phosphate. And the lipid are these little yellow tails. Notice that the phosphate are on the outside where the water is, and the lipid tails are in the center where there isn't any water. Phospholipids have water-loving heads, which are these kind of brick red color ones, and hydrophobic, which is water-hating tails, which are these little yellow structure in the center. The proteins in the membrane, which we show here as blue, act as channels for non-water molecules. You see a channel right here. Receptors for hormones. Some of these structures can be receptors and enzymes that help. For example, this peripheral membrane protein could be an enzyme that helps digest food or some other thing. There are also molecules of cholesterol, you see right here in yellow, and carbohydrates that you see sticking out of the surface here that look like these little branches of balls held together. And what that does is it helps identify the cells belonging to the self or not to the self, so your immune system can sense it and attack if it's an invader or not attack if it's part of you. Okay, selective permeability. Selective permeability means the ability to have some things move across the membrane and not others. Membranes of the cell are selectively permeable, sometimes called semi-permeable. Selectively is better. It allows some substances to cross more easily than others. They block passage of some substances altogether. For example, very large or charged ions don't move. Without this selectivity, the cell could not survive. It's what allows cells to be alive. The nucleus. The nucleus is the storehouse of information, the manager of the cell. It controls how the cell behaves. Genes in the nucleus store information necessary to produce proteins. That's the main function of the DNA and genes in our cells. This information must be copied from the DNA and then taken out to the ribosomes for protein synthesis. I'll be talking about how protein synthesis works here in just a little bit. Ribosomes then build all the cell's proteins using information sent from the nucleus in the form of RNA. But you should put a little M right here because mRNA is what actually comes from the nucleus to the ribosome to build protein. And I'll show you how that works coming up. The nucleus. The nucleus is bordered by a double membrane called the nuclear envelope. It contains chromatin, which is uncoiled chromatin. What is that? It's DNA wrapped around proteins. Makes it easier for it to condense down when you're going to do cell division, such as mitosis or meiosis. Chromatin is DNA wrapped around proteins, as I said. And inside the nucleus is a smaller structure called a nucleolus, which is different. It contains bits of RNA that are going to be used to be assembled into messenger RNA to send the message from the DNA out to the ribosomes on how to build a protein. And here's a figure that could potentially be an essay on the test. So let's take the notes here, and then you guys can fill this in. Fill it in, and then I'll talk about the figure. Okay, DNA controls the cell by transferring its coded information into messenger RNA. So here's a picture showing DNA in blue. And this is the process of converting a piece of DNA into a copy of messenger RNA. So this process is called transcription. So it makes a copy of a small section, which is coding for a particular protein. After the protein is produced, the messenger RNA is recycled back to the nucleus. So after it's all done doing its job. So let's go through how this process works. So first we make a piece of messenger RNA that's a copy of a section of DNA. Then it moves through the nuclear pore, which is a hole in the nucleus. It finds its way to a ribosome, where the ribosome reads it like a message, hence the term messenger RNA. And as it reads it, individual amino acids are put in place so that you have a functional protein. Because if even one amino acid is out of place, the entire protein is ruined. So reading it exactly, making sure that every amino acid is in its correct place is very important. And this system allows that. So if I ask you about it, tell me about this process of transcription, the making of a piece of messenger RNA, and translation, the conversion of the messenger RNA into protein. That could potentially be an essay on the test. Okay, this is the endoplasmic reticulum. This is a figure from your text. Produces an enormous variety of molecules. It can be composed of both smooth and rough. What you see here is the rough endoplasmic reticulum, and it's called rough because it has individual ribosomes studding the surface. Rough ER. The roughness of the rough ER is due to ribosomes on the outside. The functions of the rough ER include producing proteins, which is most important, and producing new membrane. So making new cells. Okay, this is a figure not from your book, but it does show you how a polypeptide is folded into a protein. The protein is put into a transport vesicle, which is a bubble, and then shipped off somewhere else in the cell, often to the Golgi. The vesicles then move to the cell surface and release the formed protein outside the cell. So this can either go directly from the rough ER and dump it, or it goes to the Golgi, which helps fold it up, finish its construction, and then dump it to the outside. Please note that not all proteins made in a cell are destined to stay there. Some are exported in this process of putting them in a bubble and exporting them to the outside. And one of the review questions says, what happens to proteins that are not destined to stay in the cell? Will they get dumped to the outside? For example, we would dump a hormone into our blood, which would travel around and do important jobs in our body. Okay, smooth ER. This is a picture showing a triglyceride, which was in a previous PowerPoint. This is a rescue inhaler. What do they have to do with each other? Well, the smooth ER lacks surface ribosomes and produces lipids, including steroids. For example, steroids could have given this guy his, big muscles. And steroids are often found in rescue inhalers for asthmatics that help relax smooth muscle in your lungs, opening it up so you can breathe better. Okay, this shows you the process of moving stuff from the protein produced on the surface of the rough ER, putting in a transport vesicle. As I said earlier, sometimes it goes into the Golgi apparatus where it gets folded up. See how it's being transported by this protein here? Eventually, when the protein gets all folded up and finished, it gets put in another transport vesicle and exported to the outside. Now this is on the surface. It can either leave or just stay on the surface of the cell. Works in partnership with the ER, which is endoplasmic reticulum. Refines, stores, and distributes the products of cells. So this is it, refining and distributing the products of this cell. Lysosomes. A lysosome is a membrane enclosed sac that contains digestive enzymes so that you can digest something inside an individual cell. What kind of cell would do that? White blood cells that engulf a bacterium or virus, or even another body cell that's died. The enzymes break down macromolecules, which are large molecules, or small cells like bacteria. So amoeba, which is a protist, can do this in the same way that white blood cells do this in our body. The enzymes may also be used to recycle damaged organelles. So for example, if mitochondria isn't working anymore, these white blood cells will surround it and, maybe not surround the individual organelle, but the organelle may fuse with a lysosome, digesting it, breaking it into bits, and then the cell makes a new one. So this is an example of a bacterium coming in through a process called phagocytosis. You see we now have a vesicle, which is sometimes called a food vesicle. The lysosome, which has digestive enzymes in it, is going to fuse with it, breaking down the bacterium, and now the cell can use those nutrients. This is a macrophage, a white blood cell found in our bodies. Lysosomes have several types of digestive functions. They fuse with food vacuoles, which is right here, to digest the food. In this case, a macrophage in your body doesn't really need it as food, but uses it to destroy the bacterium as part of our immune system. Okay, this shows you how a lysosome can fuse with a damaged organelle like a mitochondria, breaking it down and recycling it. They break down damaged organelles. All right, here's some more organelles you need to know about. Vacuoles. For example, the vacuole here inside of a plant is called a central or water vacuole. And over here is called a contractile vacuole. Contractile vacuoles, the protists remove excess water. The reason is water is constantly leaking inside of the cell because it's more salty. Water wants to go to where there's more salt. So water's coming in, and it would cause this protist to pop. So what the contractile vacuole does is it gathers up all the water that's coming inside and squirts it to the outside, kind of like bailing water out of a sinking boat. Now, the central vacuole of plants, sometimes called a water vacuole, generates what is called turgor pressure. Well, if you've ever seen a plant wilted or lettuce wilted, it's because the central vacuole doesn't have very much water in it anymore. So you can tell a plant needs more water if it's wilted. You put water on it, it's not wilted anymore because the water goes into the central vacuole, causing it to expand out, making it more crunchy. Your lettuce is more crunchy when it's been in water. When your lettuce is wilted, it's because the water has been driven out either by heat or just drying. Okay, here's a picture showing a chloroplast from your textbook. It shows you the different areas, the outer and inner membranes. You can see a thylakoid is an individual one of these green pancakes. And if you stack a whole bunch of these thylakoids together, you get what is called a granum, an individual granum. If you have several stacks, those are called grana. And the stroma is the fluid in between these stacks. Chloroplasts are the site of photosynthesis, the conversion of light energy into chemical energy. So plants and other photosynthetic protists use these organelles to make food. And this is how we have our food on this planet, because plants make food and then either animals eat it and we eat the animals, or we eat the plants themselves. Here's a picture of mitochondria. It shows something similar to the chloroplast, but instead of these discs, it shows these infoldings. And these infoldings are going to be really important in an upcoming chapter as we talk about cellular respiration. Mitochondria are the sites of cellular respiration, which involves the production of ATP. We're going to talk about why ATP is so important. It's the energy source for our entire body. Everything in our body runs on ATP. And this structure here called the mitochondria makes it. Okay, the cytoskeleton is next. Cytoskeleton is an infrastructure of the cell consisting of a network of fibers. What it does is it helps the cell stay together, changes its shape, potentially if it's a white blood cell. One function of the cytoskeleton is to provide mechanical support to the cell and maintain its shape. This is a stain showing these microtubules which make up the cytoskeleton of a cell. Cilia and flagella. Only one cell in the human body has flagella, and I'm sure you can guess what it is. It's the sperm cell, and only half of us have one, or some, which are males. Flagella propel the cell in a whip-like motion, kind of spins around this way, pushing it like a propeller. And then cilia move in a coordinated back and forth motion. So imagine all these little cilia kind of doing this, and that helps the cilia on this paramecium protist swim through the water. Okay, we have cilia too, but none on moving cells. All the cilia in us are on non-moving cells. Well, if the cells don't move, why do we have it? Well, it's on the inside of our respiratory tract. Think of the inside of your windpipe. So why do we have it? Well, we have mucus in there. What is the mucus for? So when you breathe in air containing dust, which all air has, the dust sticks to the mucus. Well, what gets rid of all that dusty mucus is these cilia on the inside of our respiratory tract, moving it up into our mouth and nose so we can spit it out or blow our nose. Some cilia or flagella extend from non-moving cells. That means the cells don't move, but the fluid on the top does. The human windpipe is lined with cilia. And what that does is it moves the mucus containing the dust that would have ended up in our lungs, but instead stuck to that sticky mucus up and out to keep our lungs clean. Okay, so that is it for this chapter. Bella, is that right, Bella? Yeah, Bella, do you have any questions about what I just covered, or do you have any questions about what's coming up on this upcoming exam for this week? Remember, it's chapters 1 through 3. It's the review questions that you've been working on. And you need to use your common sense. A couple of quick reminders. Remember, none of these are correct is almost never the right answer. But when the answer, all of these are correct, or 2 of these are correct, that's usually the right answer, almost always, as a matter of fact. So when you see it, if it was me, I would pick it. Unless you have a very, very good reason not to pick it. That's the right answer. Bella, do you have any review questions from chapters 1 through 3 that you would like to share with us, and I can help you double check your answers? Okay. Do you have any more questions before we call it a day? Okay. All right. So anyway, I hope you guys all do very well on the test. I hope to see you on Monday in class. Remember, we do have lab. You do have to come to that. So best of luck with the exam. If there's essays on it, I'll be grading it on Monday, but you'll definitely be able to find out how you did on the multiple choice. Take your time with it, and we'll see you on Monday. Bye, guys.